Hello, and thank you for joining us for the sixth annual Global Summit on Kidney Disease Innovations. Vox Populi, time is now to prevent and cure kidney diseases. The summit is hosted by American Association of Kidney Patients in partnership with our allies from the George Washington University School of Medicine and Health Sciences. My name is Vasundhara Raghavan, and I am an AAKP International Ambassador from India. I'm also a living kidney donor. I represent Kidney Warriors Foundation that mentors patients for better health and renal diet. Several books on real life instances of people with the disease have been published in the past. I'm a member of the AAKP since 2020. It allowed me to participate in actively in summits and gave me speaking opportunities that has helped me to do my work in India. It gave me an understanding of AAKP's policy making initiatives, and I recognize, as does the world, that AAKP has been the leadership in this area. This year's summit, Vox Populi, was purposely and strategically chosen by AAKP for its historical meaning, voice of the people. I'm one of the people who has been chosen to raise my voice in support of greater care, choice, and innovation in kidney care. And I'm one of the voices demanding a change in the status quo kidney medicine. Over 850 million people across the world have kidney disease. The number is increasing at an estimated rate of 5 to 7% annually. Every year, we lose thousands of innocent lives to preventable kidney issues and the lack of access to treatment. AAKP believes kidney disease is both a workforce and healthcare issue. The disease takes a devastating toll on human lives and livelihood. Most people understand that kidney disease, left undetected and untreated, can lead to premature death. But many are not aware that it impacts a person's dignity and ability to provide for themselves and their families. Kidney disease and outdated treatments can lead to unemployment, underemployment, disability, and dependency. In 2019, in 2019, AAKP declared the next 10 years the decade of the kidney. The time is now for kidney patient consumers, healthcare professionals, researchers, industry, and all the people of goodwill to unite our voices and demand change. We are optimistic we can and will save more lives. But to do this, we must demand new screening guidelines and diagnostics, new biologics, and new devices. Together, we can manage and solve this growing global public health crisis. The time is now for elected and appointed leaders worldwide to listen to patient voices. Governments must remove the regulatory and payment barriers they create that delay innovation and prevent timely care access. If government fails to act, we must raise our voices and demand an end to negative government detriments of health. Make no mistake, every nation is impacted by kidney disease and millions are at risk. The optimistic news is that every person watching this program today can help those impacted by disease and make a difference within their own nation. Through AAKP Global, AAKP is educating patients, care partners, healthcare professionals, and policy leaders across the world on unmet patient needs. Our annual Global Summit on Kidney Disease Innovations has helped put a human face on what is now the 10th leading cause of death based on data from the World Health Organization. We have connected people across the globe in a common purpose larger than one person or nation. Together, we have increased hope and optimism. So, thank you for joining us. Your voice is the voice of the people, and you can count on AAKP to keep amplifying throughout the world to create change. With that, I'm honored to serve as a moderator for our session entitled Alternatives to human organs, artificial implantable, artificial wearable, and 3D printed kidneys. Prepare yourselves for an extraordinary session. Today, we are privileged to hear from remarkable individuals who are revolutionizing the field of transplantation. You will be inspired by the pioneering work that is happening. And finally, we are going to hear from Vidmantas Sakalys. Mr. Sakalys is the CEO of Vital 3D Technologies, which uses 3D bioprinting technologies to deliver solutions for medical research, drug discovery, and regenerative medicine. Mr. Sakalys is a highly accomplished business manager with extensive experience in technology innovations across various industries such as lasers, biotech, ITT, and energy. He holds a degree in computer engineering and boasts over 20 years hands-on experience in ITT management roles, as well as over 10 years experience in phonetics innovation management. Before joining Vital 3D Technologies, he successfully established a laser research startup called Femtica. Mr. Sakalys, I turn the presentation over to you.
Thank you, AAKP. Thank you, GW University. I'm delighted here to be at this year's Global Summit. My name is Vidmantas. I will talk about 3D bioprinting and how 3D bioprinting will change the world of transplantation. Bioprinting history, what it is about, a bit of technological stuff, and what Vital 3D technologies have to do with that, and how we gonna approach bioprinting and organ transplantation issues. As you can see, and I think you understand that the organ transplantation is a really very hot topic around the world. A lot of people are just waiting for organ transplant, and just only 40% of patients are able to get this transplant in time and helping to save their lives. Really, there's a huge challenge what and how to address the organ transplantation shortage. And bioprinting is actually one of the newest concepts to help to cope with organ transplantation shortage as such. Printing itself, it's not a new concept. It's already demonstrated since 1989. The field has a lot of challenges to be solved. And between them, there is complexity of organs as such, vascularization, because you need vasculatory to feed the cells inside, cell viability, ensuring that the cells are surviving the printing process itself. Then materials and bioinks, we need to reassemble the original materials that can be very well accepted by the human body. So there is really a lot of challenges. Looking at the history itself, you can see that during these 30 plus years, there is a lot of things already printed. In this slide, you don't see the simple bioprinted organs such as bones or dental implants that does not have live tissues, because printing bones, it's much more easier than just printing the live tissues that has a vasculatory live cells and all the complexity of different types of cells mixed in it. As you can see in this slide, only the small things are printed during the history of the tissue printing. And for that is quite a lot of reasons. And we can look at the challenges we discussed in previous slide: complexity, like cell viability, ability to get the right materials. So this leads us to the next topic. technologies. So what do we use to bioprint different things? The world has discussed and has developed a lot of technologies, and some of these are simple ones like extrusion-based or inkjet-based technologies that are based on classical inkjet printers. The others are much more complex and sophisticated, such as stereolithography, laser-assisted, or even two-photon polymerization. These technologies have their own possibilities and their own challenges, but in general, the technologies are either fast or precise, but at the moment there are no single technology that is prevailing in this market, and that we can tell that this technology will be the one that we build the organ bioprinting on. Materials. We need really good materials to provide us with functional organs. In this slide, we are just talking about the materials used for laser-based bioprinting. And laser-based bioprinting uses materials that are photosensitive, meaning that they are hardening against the light. So you shine a light into the material, and this biopolymer gets harder. And on the left side, you see the smaller circle that is biopolymer. Adding to the biopolymer, mixing it with the cells, gets us bioink. So usually biopolymers are constructed in such a way that the cells are attaching themselves to the polymer molecules. So we're kind of attracted by that. That's the idea that when you will later on process the polymer and bioprint it, you will get the cells that are connected to the polymer structures. In Vital 3D Technologies, we believe that we are able to develop the technology that is meant to change bioprinting as such. So we are on a path of developing technology for printing natural size vital human organs. And this is a challenging task and brings us the very interesting results as such. The bioprinted organs that can be used for the organ transplantation. Our vision is that patient having, like for example, kidney problems arrives at the hospital, gets his analysis done, is kind of examined, gives his own stem cells, and leaves the hospital for a while. And during that period, organ is printed for him using his own cells, meaning that we will mix this person's cells with bioink and get the so-called bioink for printing the transplants. And after some time, like once or two, possibility of transplanting the organ for this specific patient will be delivered at the same hospital where he just went for examination. We believe that actually in pursuing the organ printing, the high printing speed and high precision are crucial, both of them, for the future of organ bioprinting. Actually, the challenge we are addressing is quite clear on the slide. So for the cells to stay alive in a big organ, for example, like kidney, we need to print everything very precise because, the capillary vascular network is very small, like 4 to 10 microns in diameter. That precision needs to be done at very huge speed in order to print the organ like in 24 to 48 hours, because only in kidney we can find about 100 to 150 kilometers in length of this vasculature that needs to be printed. The thing is that you are not able to print the organ for like 2 weeks because without proper nutrition, the cells will die in organ. So you need to print like 24 to 48 hours and start applying the cells inside the organ, the oxygen, start growing it. How we can do it, Vital 3D technology? So our technology uses laser-based bioprinting, and laser is a very precise instrument. We can print with laser like a pencil. We have very, very small vasculatories and other cell tissues printed. But the thing is that if you print with a pencil, like very precisely, it will be very, very slow. So for that, you need something wider, like a brush. That's where our technology comes into the play, because what we do, dynamically manipulate laser beam. We start painting the wall, for example, with a white brush. While painting the wall, we come up to the flower that we need to paint the wall. We switch to the pencil, paint the flower, and then switch back to the brush and finish painting the wall. So that's the idea of dynamic manipulation of a laser beam in order to achieve very high precision, printing smallest features of an organ, and at the same time high speed, filling the big parts of the organ with a brush. As I already mentioned, the road towards organ printing is a really multi-year strategy. And on this road, we are starting from microstructures, going to complex tissues, to vasculatory systems, combining them into complete organ for printing. So at the Vital 3D Technologies, currently we are working on scaffold organoids and vascular grafts for different drug testing and tissue regeneration experiments. Just very shortly about the Vital 3D Technology as a company. Our startup is 2.5 years old. Currently, we have 10 employees, and these employees are from very different fields: biotechnology, lasers, artificial intelligence, and precise mechanical engineering. We managed to raise 2 million euros at the moment, and looking for another 5 million in a Series A round now. We have 4 patents for the technology, and our first printer, Vital Light 3D, that you can see in a picture here, was demonstrated last year in Laser World of Photonics in Munich. Thank you, AAKP. Thank you, GW University, for having me here today, talking on this very interesting and important topic of bioprinting and organ transplantation.
Thank you, Mr. Sakalys. That was an excellent presentation. I do have a few questions I'd like you to answer for our audience. As an innovation leader in 3D printable organ technologies, how important is it for you to hear directly from patients over the burdens of kidney failure and their unmet need for new organs?
Yeah, very good question, actually. For us as leaders in developing the technology that actually can bring a lot of best things for patients with kidney disease, it's very important to understand the issues that patients are facing day to day, and that will help us to shape our technological development path to deliver the benefits as early as possible. Because in technology development, we actually can fine-tune the path in such a way that one or another benefit can be reached earlier, and for example, improved dialysis components, or some specific tissues that can help relieving the patient's pain, can be like produced earlier than just the full-size transplantable kidney. So in fact, it's very, very important to listen to the patients and to get their feedback.
Increasingly in the US, in Europe, and across the globe, government regulators and payers are asking new medical companies to show how they have incorporated patients' insights into product development. How does Vital 3D Technologies plan to do that? Will you have a patient advisory council, use survey data, or work with patient advocates?
So it's really good to have a patient insight inside the clinical and technological data. At Vital 3D Technologies, we have a scientific advisory board that is helping us with scientific strategy. And we plan to integrate at the beginning maybe one or two representatives in this scientific advisory board of a patient to help us to put the patient's insights into our development path. Later on, we are planning to have a separate patient advisory board for developing the smaller details during our clinical trials period. So we are very serious about the patients and patients' insight, and we are looking forward to work in close cooperation with patients, patient associations, and other organizations that are helping to understand the challenges of organ printing, specifically for kidney transplantation.
Looking into the future, and for the patients and policy makers watching us today, are you optimistic that 3D printing can and will make a difference in the lives of patients within this decade?
Really, that's interesting question. Actually, I am myself really optimistic that in next decade we will see the first transplantable kidney. Maybe it's not yet available for wild transplantation, but at least it will go into first trials. And actually, most of us scientists of this area agree that there is no principal obstacles in printing the transplantable organ. There is just the time and resources allocated to solve this challenge. So myself, I'm really very optimistic that we will see something already implantable during the next decade.
Thank you, Mr. Sakalys, for giving us a good insight on the important work that is being done by Vital 3D Technologies. As it relates to the impacting kidney patient care. We extend our gratitude to all of you speakers for this session, Dr. Shubho Roy, Dr. Buddy Ratner, and Vidmantas Sakalys. We are so grateful for your participation.